Life is incredible. Tiny cells make up every living thing from the smallest bacteria to the largest whale. Inside these cells, there's a special code. This code is like a set of instructions. It tells the cell what to do, how to grow, and how to work. This code is called DNA. DNA is like a long, twisted ladder. Each rung of the ladder is made of special chemicals. These chemicals are like letters in the code. The order of these letters is important. It determines everything about a living thing. This includes its traits like eye color, height, and even its risk of certain diseases. Understanding DNA and its code is key to understanding life itself. It's like unlocking a secret language that governs all living organisms. For a long time, we could only observe DNA. We could study it and try to understand it. But what if we could change the code? What if we could rewrite the instructions? This is the idea behind gene editing. Gene editing is a revolutionary technology. It allows scientists to make precise changes to DNA. It's like having a tiny pair of scissors that can cut and paste the letters of the genetic code. Imagine being able to correct errors in the DNA. These errors can cause diseases. Gene editing could potentially cure these diseases. Gene editing is a powerful tool. It has the potential to transform medicine and our understanding of life itself. To understand gene editing, we need to understand cells. Cells are the basic units of life. They are like tiny factories that make up all living things. Inside each cell, there's a nucleus. The nucleus is like the control center of the cell. It contains the DNA, the genetic instructions. DNA is made up of genes. Genes are like individual blueprints within the DNA. Each gene codes for a specific protein. Proteins are the workhorses of the cell. They do all sorts of jobs, from building tissues to fighting off infections. By changing the genes, scientists can change the proteins that the cell produces. This can have a profound impact on the cell's function and ultimately the entire organism. Evolution is the process of change in all living things over time. It's a slow and gradual process. It happens over many generations. Mutations are changes in the DNA sequence. These changes can be small, like a single letter change in the code. They can also be large, like the deletion or insertion of entire genes. Natural selection is a key driving force of evolution. It's the process by which organisms with traits better suited to their environment are more likely to survive and reproduce. Over time, these beneficial mutations accumulate in a population. This leads to the evolution of new species. Gene editing can be seen as a form of accelerated evolution. It allows us to introduce changes to the DNA more rapidly than natural processes. Section 5. Nature's editor, natural selection. Imagine a group of animals living in a snowy environment. Some animals have white fur while others have brown fur. The animals with white fur are better camouflaged in the snow. This makes them less likely to be seen by predators. Over time, the animals with white fur are more likely to survive and reproduce. They pass on their genes for white fur to their offspring. Eventually, the entire population may have white fur. This is an example of natural selection. It's a slow and gradual process, but it can lead to significant changes in the characteristics of a population over time. Gene editing can mimic natural selection in a way. It allows us to select for specific traits by directly modifying the genes. Section 6. Human hands, genetic changes, artificial selection. Humans have been manipulating the genetic makeup of plants and animals for thousands of years. We've done this through selective breeding. For example, farmers have bred crops to be more resistant to pests and diseases. They've also bred them to produce higher yields. Dog breeds are another example of artificial selection. Over centuries, humans have bred dogs for specific traits like size, temperament, and appearance. Artificial selection, like natural selection, changes the frequency of certain genes in a population. Gene editing can be seen as a more precise and rapid form of artificial selection. Section 7. CRISPR, a revolution in gene editing. CRISPR is a revolutionary gene editing technology. It's like a pair of molecular scissors that can cut DNA at specific locations. Scientists can use CRISPR to target and modify specific genes with incredible precision. This has opened up a world of possibilities for gene editing. from correcting genetic defects to developing new disease therapies. CRISPR is faster, cheaper, and more accurate than previous gene editing methods. It's a game changer in the field of genetics. CRISPR is a powerful tool. It holds immense potential for medicine, agriculture, and beyond. Section 8, the ethical frontier. Gene editing is a powerful technology. It raises important ethical considerations. One of the biggest concerns is the potential for unintended consequences. Changing one gene could have unforeseen effects on other genes or the organism as a whole. There's also the issue of consent, especially when it comes to editing the genes of future generations. The ethical implications of gene editing are complex and require careful consideration. Open and transparent discussions about these issues are crucial. As with any powerful technology, responsible use is paramount. We must proceed with caution and ensure that gene editing is used for the benefit of humanity and the planet. Section 9, the future of life's code. Gene editing is a rapidly evolving field. It holds immense promise for the future. Scientists are exploring its potential in various fields, including medicine, agriculture, and environmental science. In medicine, gene editing could be used to cure genetic diseases, develop new cancer treatments, and even enhance human capabilities. In agriculture, it could lead to crops that are more resistant to pests, diseases, and climate change. Gene editing has the potential to reshape our world in profound ways. It's a powerful tool that must be used responsibly and ethically. The future of life's code is in our hands.